We have two cell models that you will be tested on and that you'll be looking at in lab. And this is what I call the large cell model. The other one, there's another video and it's in your lab manual, is a small cell model. The reason we have two models of anything is because one model is better at some structures and the other model is better than the other one at other structures. So I first want to point out, well, first let me turn it around so you can see it in 3 dimensions. This is kind of big and unwieldy. Oh, okay. So this is the plasma membrane, the membrane around the outside of the cells. So this model is way better at the plasma membrane than the other model. And then the contents of the cell around the outside of the nucleus, this is the nucleus, this white structure. It's all called cytoplasm, and this shows you cytosol pretty well. This clearish plastic is supposed to represent the liquid portion of cytoplasm. So cytoplasm is made up of cytosol plus the organelles. And I'm not going to go over every organelle on this model. So just pay attention to the ones that I am going over, plus, of course, the list of organelles in your lab manual. So around the outside of the nucleus is in white the nuclear envelope, which is the membrane around the nucleus. If you zoom in, you can see cute little chromosomes. Well, where are they? They're those blue coiled wires. Now I feel like you could see them better. They're chromosomes versus chromatin. The other model, the small model, has chromatin. They're both DNA, just different forms of DNA. The holes here in the nuclear envelope, these are called nuclear pores. And this brown ball here is the nucleolus. Watch the spelling for that one. I don't count off for spelling. However, if you misspell nucleolus, you might accidentally spell nucleus, in which case that's wrong. This model has one nucleolus and the other model has 2 nucleoli. Surrounding the nucleus is rough endoplasmic reticulum. And then you'll also see over here some smooth endoplasmic reticulum. The white dots are ribosomes. The other model has some free-floating ribosomes in the cytoplasm. This model does not. This model only has ribosomes attached to the rough endoplasmic reticulum. This is the Golgi. These are mitochondria. Most of them are cut open. This one is cross-section versus longitudinal section. This one is intact, not cut open. And there are other things like here. I'm not going to test you on those. I'm just looking at the list rather than leaving you in silence. I just want to make sure I covered everything. And I did.